Alzheimer's represents one of the heaviest clinical burdens in modern medicine. Yet for decades, the search for a structural cure has consistently hit a wall. The research field has been divided by a 30-year schism. On one side sits the dominant amyloid cascade hypothesis, which treats protein plaques as the ultimate driver of the disease. On the other sits the vascular hypothesis, which observes that blood flow dysfunction occurs long before those plaques appear. This diagnostic scan shows the standard parenchymal view of the disease. By isolating the glowing red and yellow cortical regions, the clinical focus remains almost entirely on late-stage neurodegeneration. This late-stage focus produced the ATN biomarker framework: amyloid, tau, and neurodegeneration. For years, this rigid triad has dictated how clinical trials are designed, selecting patients based strictly on the accumulation of these three parenchymal markers. Relying exclusively on this triad yielded immense clinical trial failure rates. By targeting late-stage protein buildup, researchers were attempting to extinguish a fire long after the structure had collapsed. This framework treats the cerebral vasculature as passive pipes. In reality, every capillary forms the neurovascular unit. Endothelial cells, pericytes, and astrocytes regulate transport and clearance. Integrating this vascular axis creates the ATNV model, revealing a disease unfolding over a 50-year timeline. Across these three phases, vascular pathology systematically drives the parenchymal destruction. It conditions the initial metabolic failure in early adulthood, gates the immune collapse in midlife, and amplifies the structural disintegration in late life. The cerebrovascular system does not act as a downstream casualty of Alzheimer's. It operates as the master chronometer, dictating exactly when, where, and how the brain's defenses fail. The disease initiates between a patient's 30s and 50s in the brainstem, specifically the locus coeruleus, a densely packed nucleus of noradrenergic neurons supplying arousal to the forebrain. These cells are unusually metabolically active, requiring continuous energy from their direct capillary supply. As midlife vascular aging sets in, these tiny vessels lose their capacity to autoregulate. The result is chronic capillary hypoperfusion and early compromise of the blood-brain barrier at the vasa nervorum. Because the locus coeruleus relies on a single arterial source, this hypoperfusion causes a sharp, localized drop in oxygen delivery, right at the site of the brain's highest metabolic demand. Oxygen starvation triggers severe mitochondrial oxidative stress. To repair the DNA damage, the neuron hyperactivates a repair enzyme called PARP1. Sustained PARP1 activity consumes massive amounts of NAD+. This molecule is the essential currency of cellular energy, and its rapid depletion shuts down the cell's remaining metabolic machinery. The parenchymal cell fails entirely because the upstream vascular supply could no longer meet its baseline physiological requirements. The locus coeruleus neuron does not spontaneously die of old age. Its vascular supply slowly degrades until the cell suffocates on its own unmanaged metabolic exhaust. The sequence progresses into the 5th through 7th decades. The site of failure moves upward from the deep brainstem into the temporal lobe. The focal point for this second phase is the hippocampus, the critical anatomical engine for memory formation. The hippocampus harbors a unique architectural vulnerability. To support rapid neurogenesis and continuous plasticity, its baseline vascular network is constitutively leakier than the rest of the cortex. Over decades, systemic inflammation accumulates in the blood plasma. This aged systemic milieu places constant, aggressive pressure against the already porous hippocampal barrier. The endothelial cells react to this toxic plasma by inducing a specific protein. They physically express vascular cell adhesion molecule 1, or VCAM1, sprouting receptors into the bloodstream. Circulating aged monocytes then use specific integrins to dock tightly onto these newly formed receptors. This physical binding triggers a localized reactive oxygen pulse. In response, the endothelial cells secrete inflammatory cytokines, specifically IL-6 and CCL2, downward into the brain parenchyma.
These cytokines diffuse directly into the brain tissue until they reach the local microglia. The inflammatory signal overrides the surrounding maintenance cues, permanently destabilizing the microglia's homeostatic signature. Stripped of their protective identity, the microglia physically transition from passive sentinels into an active, destructive, disease-associated state. Systemic aging does not passively seep through a broken barrier. It actively weaponizes the endothelial surface to corrupt and reprogram the brain's local immune system. Moving into the 7th decade and beyond, the pathology reaches the outer cortical microcircuit. Soluble waste normally flushes out along perivascular pathways. As clearance fails, amyloid beta-40 peptides become trapped. They embed deeply into the vessel walls, a condition called cerebral amyloid angiopathy. As these amyloid deposits physically replace the healthy smooth muscle cells, the vessels become rigid and brittle. This structural decay paralyzes the arterioles, destroying their capacity to regulate blood flow and compounding the brain's baseline hypoperfusion. Meanwhile, the corrupted microglia generated during phase 2 begin flooding the cortical tissue with an aggressive digestive enzyme known as MMM9. The primary target of this enzyme is the parvalbumin perineuronal net. This dense matrix physically shields the fast-spiking interneurons that synchronize brain activity. Under the flood of MMM9, the nets are rapidly digested. Stripped of their protective and metabolic scaffolding, the interneurons misfire. The precisely timed gamma-frequency computation required for complex thought simply disintegrates. MMM9 also has a simultaneous secondary target. While it digests the neuronal nets, it actively degrades the basement membrane of the surrounding blood vessels. The immune dysfunction attacks the neuronal scaffolding and the vascular scaffolding at the exact same time, creating a catastrophic amplification loop. The severe cognitive decline seen in patients in their 70s is not a sudden, isolated event. It is the structural convergence of 40 years of vascular and immune decay, completely digesting the brain's physical architecture. Stepping back, we observe a continuous 50-year cascade from the locus coeruleus through the hippocampus to the cortex. Genetic risk factors amplify this sequence. The ApoE4 variant operates as a constant multiplier, accelerating pericyte degeneration and vascular damage across all 3 distinct phases. Simultaneously, age-related disruptions in slow-wave sleep diminish glymphatic clearance. The brain's natural ability to flush waste drops year after year, applying continuous toxic pressure to the entire system. For decades, the pharmaceutical industry attempted to arrest this vast, interconnected decline with single-target drugs aimed exclusively at late-stage protein plaques. The biology demands a different approach entirely: developmental pharmacology. Physicians must intervene on the exact biological substrate that is actively failing in a specific decade of life. During the phase 1 window in a patient's 30s and 40s, the necessary intervention is strict cardiovascular risk control, maximizing blood perfusion to prevent the brainstem's bioenergetic ignition entirely. In the phase 2 window of late midlife, the target shifts to the blood-brain barrier. Pharmacological VCAM1 blockade could effectively seal the microglial gateway before systemic plasma corrupts the local immune system. By phase 3 in a patient's 70s, the available intervention is administering anti-amyloid monoclonal antibodies, like lecanemab, to physically strip plaques out of the cortex. However, this late-stage strategy hits a severe vascular constraint known as ARIA. Because phase 3 vessels are structurally compromised by amyloid angiopathy, using antibodies to pull amyloid out of the walls frequently triggers edema and cerebral microhemorrhaging. We cannot medically extract late-stage amyloid without accounting for the highly degraded vascular scaffolding that must survive the clearance process. Alzheimer's disease is a lifelong vascular cascade, and neurology must finally treat it as one.